Hey guys, welcome back to our channel. So today, Nina Simone and I are going to be talking about parthenogenesis. And I know you're probably like, what the heck is that? So stick around to find out more. Hey guys. So if you've made it this far, then you're probably a bit curious about what parthenogenesis is, and I'm here to tell you. So parthenogenesis is a form of asexual natural reproduction by which embryos develop from unfertilized eggs. There are two types of parthenogenesis, and these two types occur from two different methods of automixes. On the left, you see our first type, which is obligatory parthenogenesis. This form of parthenogenesis occurs in species where asexual reproduction is the exclusive form of reproduction, and it occurs by necessity. Obligatory parthenogenesis is rare, so it's not as common, and it's usually found in all female populations. At the bottom left here, you see this little guy, kind of looks like an earthworm, but this is actually a Brahminy blind snake. So this species of snake uses an apomictic form of parthenogenesis in which offspring develops directly from mitotic divisions of unreduced ova, which are mature female reproductive cells. On the right here, we have our second type of parthenogenesis, which is facultative parthenogenesis. And it occurs in populations of species that reproduce sexually. And so therefore, parthenogenesis is not as common. Facultative parthenogenesis is kind of a last resort for females to reproduce asexually, kind of as a last resort because they're having trouble finding mates. So at the bottom right here, you see the cottonmouth and copperhead snake, which typically partake in this form of parthenogenesis. So if you've made it this far, then you're probably... So here we have a visual representation of the multiple forms of reproduction that we have discussed thus far. So on the far left, we have a blue and a red fish. The blue represents a male fish, the red represents a red fish. These two fish mate and produce both male and female offspring. This is a prime example of sexual reproduction. So moving towards the right, we have a red fish producing another red fish. So these are both female. This female fish is present in a species that contains sexual reproduction as their main form of reproduction, but for reasons unknown, possibly because she was unable to find a mate or something of the sort, she did undergo asexual reproduction, specifically facultative parthenogenesis, and only produced offspring with her DNA, with female DNA. All offspring that are produced as a result of parthenogenesis, both types of parthenogenesis, only contain DNA of the mother, of the female. So looking towards the right, we have an example of obligatory parthenogenesis. This female is part of an all-female population, so she underwent obligatory parthenogenesis, asexual reproduction, and she produced all-female offspring containing her DNA. So here we have another visual illustration of facultative parthenogenesis automixes. And I kind of mentioned that term automixes earlier and kind of just skimmed past it. I know you're probably like, huh? Here I am kind of explaining that in further detail so we're on the same page. Automix is essentially the process where two haploid gametes from the same meiosis combine. When the region around the centromere is completely heterozygous, this indicates that central fusion has occurred. When the region around the centromere is completely homozygous, this indicates that terminal fusion has occurred. Central fusion automixes result in haploid gametes with different combinations from the female parent. Terminal fusion automixes is haploid gametes with the same combination pattern as the female parent. And in both instances of automixes, the resulting offspring are half clones of the single female parent. Because, like I mentioned before, all offspring of parthenogenesis contain the DNA of the female. It's just all of her DNA. Here we have another. So really quickly, I kind of just want to go over a brief background of what we found in our research analyzing articles and things of this sort. So we found that parthenogenesis was believed to originally be a circumstance of captivity because the first cases were observed in captive snakes in zoos around the world. So following studies on parthenogenesis, we found that this mode of reproduction was prominent in populations where the population number was low or where males were not present at all. Thus, this method of reproduction was thought to be kind of a last resort. So on the left, we have an article entitled Parthenogenic Zoo Burmese Python Snake. And in this case, a female Burmese python snake reared in captivity at the Artis Zoo of Amsterdam in the Netherlands, was in isolation from males. So she produced annual clutches of eggs between this 5 years, between 1997 to 2002, of which 25 to 30% of her clutch appeared to contain healthy embryos. So on the right, we have our second study entitled Parthenogenic Small-Toothed Sawfish, which kind of basically discusses that a critically endangered rare species that was estimated to have declined between 1 to 5% of its population size in 1900. Basically, the sawfish parthenogens, were normal sized for up to 1-year-old individuals, thus demonstrating that individuals produced in this way can survive in the wild. So really quickly,
Hi, my name is Denise Simone Warner, and I'm going to be continuing this presentation. So, in the research on parthenogenesis on North American pit vipers, Warren and other researchers used microsatellite DNA to do the analysis to test if copperheads in cottonmouths are capable of facultative parthenogenesis, or if the offspring are a result of other things like sperm storing or incest. So the researchers found that no other individuals except for the mother and the offspring had genotypes that were compatible with each other across all loci homozygosity. Combined with litter characteristics like a male-only offspring and high numbers of developmental issues, this represents the first case of facultative parthenogenesis in wild vertebrates. This quote down here is explaining that the researchers thought that because parthenogenesis was thought to be something like a last resort, the researchers thought they would find the same thing here. However, it's not looking like it's a last resort because there were definitely a lot of males on the population, and this parthenogenesis usually shouldn't happen in populations where there are a lot of males. And in the population with cottonmouths and copperheads, there is a skew towards a male dominant population. So the females definitely didn't have an issue finding males. However, the researchers are hypothesizing that the females didn't want to mate with the males present because they couldn't find their one true love, their like really suitable mate. So they decided to do parthenogenesis. And this is just an example of what I was talking about earlier about homozygosity across all microsatellite loci. So there's 8 loci for the cottonmouth and 7 for the cottonmouth. No, 8 for the cottonmouth and 7 for the copperhead that are indicating homozygosity across all loci. And as you can see here, the offspring and the mother have a lot of things in common across all genotypes here, and that's usually something that's very common in parthenogens. So the offspring from this research, the parthenogens, the cotton mouse had no apparent health issues. They were released after their birth, but the copperhead species, they were kept in the zoo, and they are still living to this day. They had no issues with growth. They had no issues with eating, and they grew up to be really healthy. So this slide is about parthenogenesis on Australian elapid snakes. Researchers Allen and others documented the first observation of facultative parthenogenesis in the family Elapidae on two snake species, the coastal taipan and the southern death adder. This study was done on 4 females, 3 of which were the coastal taipan and the other one was the southern death adder. So the out of the 3 coastal taipans, 2 were raised in captivity at the University of Adelaide, and the other coastal taipan was caught from the wild for this study. And the southern death adder, which was the only southern death adder in this research, was a wild caught specimen as well. So similar to the other study, since these snakes usually have males in their population, and these snakes were, the experiment was done inside the university, so it wasn't done like out in the wild. They wanted to like do mocks, or they wanted to basically mock like males in breeding season. So they brought males in for 1 to 5 day periods during mating season to try to get the females to mate with them, to prove that parthenogenesis isn't because of a low population of males. However, it's due to the female's choice. Oh, was I speaking? I think so. I'll look later because it says mute. LOL. So all the females had the chance to reproduce sexually, but they chose not to. And in this study, the females did reproduce sexually previous to having parthenogenic children, which means that this sperm, that these offspring weren't a result of stored sperm, and they most likely were not a product of incest or inbreeding. So here is an explanation of what I was talking about earlier across the loci, that this is definitely a result of parthenogenesis. So there are 3 genetic tests done to ensure that this is parthenogenesis: and an internal relatedness test, a standardized heterozygosity test, and a homozygosity test across all loci. So a high interrelatedness greater than 15% is significantly greater than 0. So that means that the mother and the offspring are very much related. And then for standardized heterozygosity, if it's low, generally under 50%, and then combined with homozygosity across all loci being high, above 60%, then we're looking at parthenogenesis. And then this is a result of the offspring after being born, with the 4 females that they did research on. And we can see here that there were some like slight defects and infertile snakes that were born. But all of the coastal taipans that were alive at the time of the publication lived for a really good time. They were healthy and didn't have a lot of abnormalities that were inhibiting their growth. There were 2 that had like slight abnormalities, like a deformed eye or having malformed scales, but the other 2 had no abnormalities or issues with their health. However, on the other side, the one southern death adder that was included in this study did have southern death adders down here. They did have like issues with their health. They did have defects, and they died less than 2 months after their birth. So the first snake that we see here on the right is a coastal taipan, and the one on the bottom is a southern death adder, just for like visualization. So in conclusion, why parthenogenesis? Researchers still don't know, because other species use parthenogenesis as a means to repopulate their to repopulate their population when the population is low, when there's no males. However, we're not seeing this trend in these researching on snakes. We're not seeing this trend. Both snakes in the family of the American pit viper and the Australian lapidae did have the opportunity to mate with males. However, they didn't. And in American pit viper populations, they're usually heavily male-based populations, so the probability that the females aren't coming across the males is very, very low. So more research should be done on parthenogenesis on snakes, and the reason that females choose to do parthenogenesis and the outcome that it has on the population, because right now we are not exactly sure. We have no reason at all. But thank you for listening. I'm Nina Simone Warner, and have a good day.